Hello, welcome to this first in a series of videos that are designed to help users of the SumLine breast cancer cell line knowledge base use the website more effectively and efficiently and make best use of all of the data mining tools that we currently have on the website. I know at this point there's an awful lot of content that we've put on this knowledge base and website, and it might be a little intimidating or a little overwhelming when you first come to it, particularly the way we've tried to organize it. So I want to have a series of videos now to sort of take you through each piece of this in an attempt to make this all make a lot more sense. And because I really would like more people to be using the data mining tools. I think it's been encouraging that our membership to the knowledge base has grown significantly over the last few months. But I get the sense that not a lot of people are making great use of the data mining tools. And that really was one of the main purposes of developing this resource. So today in this first video, I'm just going to go over the overall layout of the website and how it's organized. Because again, there's so much content, there's so many pages that it can be a little difficult to follow. So we'll start with that. And then after that, we'll do a series of individual videos to take you through each of the data mining tools currently on the site. Okay, so I'm going to go ahead and share my screen now, and we'll talk about this overall organization. Okay, so this is the front page of the SUM breast cancer cell line knowledge base, as many of you are aware. And this is the page you come to when you first find the website. And obviously it talks about how you need to get a password and so forth. The idea when I sort of laid it out this way was after people would come to the first page, scroll down to the bottom, they'd come to this Let's Get Started link, and that would take them immediately to this first real content page of the website, the so-called SLKBase page. So here is really the summary of everything that's on the website and how it was designed and what we're trying to accomplish. So I hope everybody would just take a little bit of time and just read through all of this, because really this is meant to be the launching point for the knowledge base. But one of the things I want to just show you here today on this first page is what's written over here in this margin, because this really talks about the data that we put in the knowledge base and in the MySQL database that all of the data mining tools leverage. And so it's really important that everybody sort of understands what was the starting point for all of this. So within our database, we have these various data sets for now about 70 breast cancer cell lines. So first of all, copy number data, and it's actually for candidate oncogenes, but it's really copy number data from genome-wide array CGH analysis. So that's all in the database. Point mutation data derived from exome sequencing, genome-wide expression data from RNA-seq analysis, and genome scale shRNA and CRISPR screen data now for all of the cell lines. And together, all of these data sets really form the core of the MySQL database, and we try and leverage all of that within the content of the data mining tool. So, so please be familiar with that. And then again, there are some key links here, key features of the breast cancer cell line panels here. For people who have come to this website because they're working with the SUM lines, we have pages on the media and protocols and so forth. Okay, so again, a lot of key sort of introductory information in this first page. What I really want to show you today, again, is more about how this website is organized. So again, we started this website to be focused on the SUM lines, and then later expanded it to 50 or so additional breast cancer cell lines. So these first pages up here in the banner really are focused on each of the SUM lines, and you'll see that each some breast cancer cell line has its own homepage. And then what you'll see for each of these home pages is that when you mouse over one of them, a bunch of drop-down menus will drop down. So if you actually just click on the Sum44 homepage, for example, you'll see these other pages that are sort of all within the Sum44 home, a narrative summary about how the cell line was derived, some published papers from the Sum44 cells, a narrative summary about the sort of the biological characteristics of this cell line, some information about oncogenes and point mutations and some common KEGG pathways that are enriched with data from the screen that are pertinent to the Sum44 line, and some druggable signature kind of data as well. So at some point as we go through these videos, we're going to get in depth to all of these, but all I want basically you to see at this point is that for all of these pages that are relevant to the Sum44 breast cancer cell line, there's basically two ways to get to them. You can just come to this homepage, and then you can just click on any one of these pages and read the story of Sum44, and then go back if you'd like, and then come to the next page. The narrative summary of the line is you get through this way, and it's sort of a fun read if you're interested in the biological characteristics of this cell line. So you can do it this way, or again, the way this is organized, you can simply mouse over, and then all of these are the same pages, right? So oncogenes, candidate oncogenes, functional druggable signature. You can see that these pages are all the same as these pages. And so there's basically, so all of the lines are laid out that way. So the easy way to get to all this information is simply to mouse over each of the cell lines. And then if you want to see the derivation of the Sum52 breast cancer cell line, you can get to it this way. And you can either then go back to the home page or just mouse over it again. And if you want to see the narrative summary of the 52 line, you can do it that way as well. So again, this is meant to make navigation of the website a little bit easier, because again, I think at some point, at this point now, we have roughly 300 pages of content on this website pertinent to all of these breast cancer cell lines. So each of the sum lines has a home page like this. They all work in the same way. You can use these drop-down menus to get to any one of these sub pages that are just packed with information about each of the sum breast cancer cell lines. Okay, so that's how we started this, and there's a lot of, again, very interesting information, but it's information basically, and so what we wanted to do to get beyond sort of just information is provide some ways for people to dig down into the data that we have and use some data mining tools to make sense of that. So what you're going to see in this banner is we basically have three different pages that have data mining tools. So this first one is the first one that we developed, and these are the data mining tools for the sum breast cancer cell lines only. And then these are the data mining tools for the additional 50 breast cancer cell lines. Now you'll notice that the tools themselves are essentially the same. In each case, we have an oncogene signature tool, a KEGG pathway engine, a gene query tool, and so forth. And pretty much all of those tools are present here for the 50 additional cell lines. So you may be wondering why we've separated these out into these two different parts of the knowledge base. And the reason for that is actually pretty straightforward. The reason for that is that because all of these data mining tools make use of the functional screen data to try and functionalize all of the other genomic data sets, we actually have a couple different sets of genome screen data. So we performed the genome-wide screen in my laboratory, and we did it for the sum breast cancer cell lines. And we did it for MCF10A cells, MCF7 cells, and MCF7 long-term estrogen-deprived cells. And, we did that using our published method for an shRNA screen. And in fact, if you want to see in detail how we performed our screen and how we generated the data we did, you can find that by clicking on these links from that SLKBase page, how the shRNA screens were performed. And there's also a tutorial on the KEGG pathway engine, but we're going to redo that one actually shortly. So again, if you want to see how we did the screens, you can click here and see a PowerPoint presentation or actually see a video presentation of how we did the screen. So again, all of these tools for the some breast cancer cell lines, as well as MCF7, MCF10, MCF7 LTED cells, were performed using our select a screen with our statistical method for analyzing the screen hit data to determine which genes we consider to be hits in the screen and which that we didn't. And again, all of that data are leveraged within these tools. For the data mining tools for the additional 50 breast cancer cell lines, what you can see here is that all of the data that is present in our database for these additional lines come from this site, DepMap Portal, which is the Bill Hahn site at Dana-Farber. And basically, what we did is we downloaded all of their data on copy number, gene expression, point mutation, and the results of their genome scale shRNA screen, some of which were done with RNA interference-based methods, some of which were done with CRISPR methods. We downloaded all of that data into our database and then used exactly the same software and exactly the same tools to run our data mining tools shown here. So because the computational and statistical methods that were used to analyze the screen hit data from the Hahn group and our group are different, we didn't really feel like we could merge all that data together. And so for that reason, these two groups of cell lines are done separately. But then as you'll see, because the Hahn group did some of the cell lines, there's some overlap there, which is actually pretty helpful. And it actually made me feel confident that, that there was pretty good overlap in the data that they generated from our cell lines and the data that we generate. So that's the first thing I wanted to be sure everyone understands, is why there are these two different parts of the website that contain data mining tools. The tools are the same. In the case of the SumLine data mining tools, all of that genomic data were generated in our lab. We did the gene amplification, gene deletion data, we did the exome sequencing, we did the RNA-seq, et cetera, whereas all of the data, the other 50 lines, were done in other labs, predominantly Bill Hahn's group at Harvard. The other thing I just want to show you is the proteomic analysis part of the website. At the present time, we only have the proteomic data for the sum lines. Now, as we're working toward continuing to expand our knowledge base, we will incorporate RPPA data and have proteomic analysis for the other 50 lines as well, but we don't have that at the present time. Again, in the future video, we'll spend the whole time talking about how to use this proteomic analysis tool. I just wanted everybody to understand at the outset that this tool currently only applies to data from the some breast cancer cell lines and not any of the others. Okay, so let's get back to the data mining tools themselves. So we have, again, several different tools that are common between the two. This oncogene signature tool, in which you'll be able to identify the driving and candidate oncogenes operative for each cell line. The KEGG pathway engine tool, and in this case, we have a KEGG pathway analysis and druggable signature tool, which I'll go into in detail in separate videos. And then a gene query tool, which I think is the most fun and the easiest to use and the most informative for most people. And again, a tool for proteomic and phosphoproteomic analysis of our data set. So that's all present here for the sum lines. And then for the other 50 cell lines, we're working toward having the same tools, but we certainly currently have the oncogene signature tool, the KEGG pathway engine, the gene query tool, and the functional druggable signature tool. Now, in addition to that, I want to point out this page, the SLKBase blog, where there are a number of blog posts, and I'm currently in the process of writing a number of new ones that will be posted. And these blog posts are going to be designed to help help you understand sort of the background and the basis for the analysis that can be done with the data mining tool. So, for example, I mentioned that in this data mining tools for the breast cancer cell lines, these data come from the Han Lab, and the Han Lab did both RNAi and CRISPR. You can see here KEGG pathway data for CRISPR and RNAi data. And so there's a blog that I'm getting ready to post shortly, which really talks about those two different data sets, the similarities, the differences, and how to sort of interpret some of those differences. So I'm getting ready to post a blog on that. If you're watching this video, a few days or weeks from now, you'll notice that blog is already present. Please take a look at it. But I've got 3 blogs here currently, and these are just sort of informational, they were fun to write and I think hopefully fun to read. But the one that I think everyone ought to read is this blog here called Cell Lines Are People Too, because that really is where I'm trying to make the case that breast cancer cell lines are not just research reagents, but in fact are stable and very powerful models of the individual disease or the particular disease that the individual patient had that gave rise to each cell line. This is a clearly important premise for everything that's going to come in the future as we develop this knowledge base further, because in order to continue to get the most out of what we're trying to accomplish here, you have to be on board with this idea that cell lines are people too. And if you're not, then you're not going to believe or take seriously a lot of the stuff that's to come. But I think if you read this carefully, I think at least we start to make the case that we all should be thinking about cell lines a little bit differently than we currently are. And I sort of go into the pros and cons. So please read that when you have a chance. Okay, so I just wanted to take this first video, this short video, to just be sure everyone is understanding of how the website is laid out, and with the idea that you can use these drop-down menus to easily navigate through all parts of the website, and you can get to all of this information very, very quickly and very, very easily. And I think when you sort of get this overall organizational scheme, it helps navigate the site and allows people to, again, very quickly get to the information that they want to have. Okay, I'm going to stop my screen share at this point. And again, thank you for watching this first video. I hope it lays the groundwork for the ones to come. And, I think if you explore the website and begin to work with some of the data mining tools, whether it's for cell lines you actually normally work with, or if it's for cell lines you haven't worked with, but are of interest to cell lines you have, I think you'll find surprises. You will learn things about the cells you work with that you never knew. I guarantee it, because I still learn about cells that I work with using these data mining tools. So I hope that's a good teaser, and will convince you to start spending a little bit more time with this knowledge base. So again, thanks for tuning in, and I will see you next time.